There has been a great deal of media coverage about stem cells and their potential as new therapies. How much of this is true? Journalist and commentator Quentin Cooper chairs a panel discussion on the future of stem cells at the Newcastle Science Festival.
Thank you very much. Given this event is called The Future of Stem Cells, I thought we'd start by going back to the future with something from Michael J. Fox, who of course was the star of the Back to the Future trilogy and was diagnosed with Parkinson's nearly 20 years ago, one of many diseases which it's thought or hoped stem cell research could one day help cure. In a recent US interview, he said, and I'm not going to try the accent, "A vast majority of people in this country are in favour of science playing a leading role in making changes in the future and believe in stem cell research. We've decided we'd like to take this step, to do it with caution, with the strictest adherence to ethics and all of the principles this country stands for, but allow us to do that without infusing the conversation with inflammatory rhetoric and name-calling and fear-mongering. It doesn't help." It doesn't. And tonight we're going to try and diffuse any inflammatory rhetoric and any hype and look at not just the kind of Hollywood image of the possibilities as to what stem cells can deliver, or the anti-stem cell fear-mongering image as to what stem cells can't deliver and shouldn't be allowed to deliver, but actually the realities and how we actually achieve the best possible from the potential that's there. Just in the last year or so, not only have there been major advances in areas such as reprogramming skin cells to become stem cells, there's also been a change in the research climate, the good kind of climate change, and the kind that Michael J. Fox was on about when he was trying to encourage more research in the United States. Now that should all be clear, but just in case it isn't, you can always follow the pretty pictures. In case you've been wondering, standing to my extreme right over here is Nick Payne, who is our sort of artist in residence for this event. And I think, Nick, we should just explain briefly what you're doing.
Tonight I am going to be attempting to record the discussion in a kind of visual form on this enormous and at the moment worryingly blank piece of paper.
Are we all clear? Ish? Thank you. Right, well then let's start. We're going to hear first from Professor John Byrne from West Auckland. He's now a drummer in covers band Famous Last Words, but slightly more relevantly tonight, he's medical director and head of the Institute of Human Genetics at Newcastle University. Professor Sir John Byrne.
Thank you, Quentin. Ladies and gentlemen, it's a pleasure to be here. The reason that I became involved in stem cells was actually about 10 years ago. I was asked by Liam Donaldson to take part in his report, the Donaldson Report on stem cell research. So when I sat on this committee with Liam and a whole lot of people, theologists, philosophers, etc., we talked about what was special about stem cells. And we decided actually there weren't any really new ethical problems to be confronted. And that comes as a surprise still to many people. But what had been discovered about in the late 1990s was that if you took a very early human embryo, which was still at the very earliest stage of just a ball of cells, it hadn't divided or developed into anything, and took any one of those cells under the right conditions in the laboratory, you could persuade them to remain stem cells. They didn't differentiate and turn into something. They just sat there doing the same thing. And that meant that the scientists who worked with this could then begin to study what the control steps were that would make them move on to become liver or bone or heart or whatever. The other thing that, of course, caught all our attention was Dolly the sheep. The ability to actually manufacture, if you like, a mammalian clone, which was obviously very close to us humans. And what the team in the Roslin did was they took a nucleus and they stuck it into an embryonic stem cell, and they then made that into, or set that off to grow. And what it was effectively a genetically identical product of Dolly. And what that meant was that we could see into the future the possibility of transferring a nucleus from an adult into one of these stem cells, growing it up, and then potentially making replacement parts. And that obviously captured the imagination both positively and negatively. Subsequent to that, a number of people began to work in our centre and elsewhere around the world on other types of stem cells, particularly cord blood stem cells, which fall somewhere in between, because In the cord, there are cells which are primitive bone marrow cells. And what was even more amazing was that the baby's blood, the cord blood that we can get from a newborn baby, contains cells which retain that marrow potential. They can actually do the same thing. And so the question was, could people actually take those so-called adult stem cells and turn them back to behave as if they were those very early apprentices, those embryonic stem cells? And great success has been seen in the last few years with the ability to stick a few genes in and really do that, to persuade these cells to go backwards and to be induced pluripotential cells. And so the big debate now is whether in fact we can scale up those induced cells and persuade them to do what we want them to do, or whether we're going to need to use human embryonic cells because they're the only ones that can really do the job properly. They haven't already begun to differentiate. And so a lot of our scientific debate is about whether we can control and understand those processes. Meanwhile, of course, there are all sorts of downstream debates about things like whether we can scale them up, but there are also continuing debates about where the ethics fits into this. Is it reasonable to use unwanted embryos from the fertility service to grow these cells? Is it indeed necessary? Or can we actually dispense with that entirely and just focus on the induced pluripotential cells or cord cells, etc.? My feeling is that the human embryonic cells will remain part of the story because they're essentially the gold standard. If we can really understand what makes them tick, then we will gain the knowledge that will then allow us to properly manipulate the adult stem cells. But the bottom line is that in here somewhere is the magical treatments that we all aspire to. In here is the potential to treat the patient with Parkinson's disease or the patients with diabetes. And it's because that potential exists that I and other doctors are interested in pursuing this field. It might turn into sand, it might not work, but my strong suspicion is that it will. And diseases that we previously considered incurable will have started to yield to medical intervention. Thank you.
Professor John Bird, thank you. Our second, or depending on your point of view, penultimate speaker is Dr. Ruth McKernan. Among many other things, she's a science writer, author of the Ventis Prize shortlisted Billy's Halo, which is about her father, but does have a chapter in it on stem cells, and also Chief Scientific Officer with Pfizer Regenerative Medicine. Dr. Ruth McKernan.
Thank you. So thank you, and it's my pleasure actually to really extend what you've said, John, and talk about some of the therapies that are available, will be coming available, some of them soon, some of them much further in the future. As you said, there are niches, there are parts of the body that still retain stem cells as we grow and develop all the way through life. The bone marrow is probably the one that you're most familiar with, but fat is also a good source of stem cells, and actually there are stem cells in the brain. There are some in the heart that are able to regenerate parts of the heart muscle, and I would say the only organ that we're pretty confident doesn't have any is the pancreas, and that may be one of the reasons why actually diabetes is really quite a high incidence disease. Because stem cells from the bone marrow can only make the sorts of cells that bone marrow normally makes, they can only really be used for therapies that would use those cells. So bone marrow can make cartilage, it can make bone, it can make fat. So, for example, you can take bone marrow cells, culture them, and use them for cartilage disease, for people that have really lost a lot of their cartilage, and that is available now. There's a therapy called ChondroSelect that you can have in countries where it's available at quite a considerable price, but that is a cell-based therapy that's already available. When it comes to regenerating whole organs, if those are organs that are based on the cells that come from the bone marrow, it's possible that there's a future there. We can think about using bone marrow in two different ways. Either I can use it for my own cells, that would be an autologous therapy, or we could take those cells and expand them as far as is possible and use them to treat many people, and that's called an allogeneic therapy. So for my last area, human embryonic stem cells, these are the cells that John talked about that are infinitely expandable. They are so expandable that one embryonic stem cell can make a line that can be used to treat the entire world's population of macular degeneration. And that's one of the areas that we're working on. So we have a collaboration with University College London, some really smart guys there, Professor Peter Coffey and Lyndon de Cruz, who've worked out how to make part of the eye, the retinal pigment epithelium, how to make that from an embryonic stem cell line. And what we're doing with them is growing those cells on a little disc, and that disc can be put behind the macula of people with very late stage macular degeneration. Now, we've been working on this for about a year, and if all goes well, and my experience in drug discovery is actually things always happen that you don't expect, but if all goes well, we'll be able to start clinical trials on patients next year. So that's an embryonic stem cell source that we plan to use for the treatment of macular degeneration. But I think we can really look forward to a lot of different types of stem cell therapies. Bone marrow-derived therapies first, macular degeneration next, pancreas diseases after that, and I think neuroscience is the hard slog. Thank you very much.